And thank you so very much for the invitation. Why do we sleep? We spend essentially 1/3 of our lives sleeping. Essentially all living creatures sleep. And in the words of William Shakespeare, we are such stuff as dreams are made of. So why do we sleep? One idea is that we sleep in order for the brain to have a chance to clean up. This hypothesis hit the news with a bang via a 2013 science paper from the group of Mike and Edgar in Rochester. And what they did is that they injected, in a series of experiments, injected tracer into the fluid surrounding the brains of mice. And they observed that this tracer, this fluorescent dye or contrast agent, spread into the brains of sleeping mice much, much faster than in awake mice. So the sleeping mice here is the green and the awake mice is the orange. They also found that the tracer was cleared faster in the sleeping mice than in the awake mice by a factor of about 2. So this raises a number of questions, like how is this even possible? And what we're interested in addressing is try to quantify the biophysical mechanisms at the macro scale that could underlie sleep, that enable transport that they observe in sleep. Now we are very, very far from the first look at this. You know, these are questions that have been around for decades, probably 2 centuries. We've been looking at this for 5, 6, 7 years now using mathematical modeling. And I think that we now have some intriguing new answers to these classical and some new quantifications to these classical questions. So let's start by taking a step up on the evolutionary ladder. What you're seeing here are 2 rows of MR images in humans. The rows are from different views, and what you see are different time points. So the leftmost one is a baseline. Then you have after 1 to 2 hours, between 4 and 7 hours, after 24 hours. We also have data after 48 hours and after 4 weeks. These are MR examinations, so-called contrast MR examinations of patients in which the protocol is that they inject this contrast agent into the fluid surrounding your spinal cord, or these patients' spinal cord, and then they roll these patients in and out of the MR machine and image how the tracer spreads in the human brain. So the question now is, how does it spread? We see that it spreads into the brain, but how does it? Now this is contrast agent is gadolinium, and it's assumed not to a substantial effect cross the blood-brain barrier, so it does not spread via the blood. So how does it spread? Well, these clearly, what you can also see here is that it seems to spread from the surface, and this tracer is injected in the fluid surrounding the brain. So could it be spreading just by diffusion? And really now we're starting to kind of venture into a point of discussion and a field discussion where what are the modes of transport into and out of the human brain has been around again for decades, probably centuries. But we're going to start investigating this. And it could be, first, the most thing you could think of would be that it spreads by diffusion alone. Now, this is something we can model. So what we did was that we considered subject-specific kind of macro-scale brain models of diffusion of solutes into the brain. So we have a cohort of around 20 patients. And for these, we have 2 structural MR images so that we can create subject-specific geometries of the brain. We also have contrast data at certain time points. These are kind of, as you can see from the previous image, we don't have that many time points, so they're low resolution in time, but relatively high resolution in space. And from these concentrations, we can map them onto, from the single intensities, we can map these onto concentrations in the brain. And we can generate subject-specific brain meshes and numerical models of this. And we can then assume that, okay, what if diffusion is the only physics that's going on here? What would the predictions then be? That what we do by solving this diffusion equation, and what we say is that we prescribe the concentration that are imaged on the boundary alone, and then say, okay, if it's only diffusion that happens inside, what would then happen? And let's look at what we found. Well, we're quantifying what we find by looking at the total amount of tracer in the white matter and the gray matter. And what we find is that if you only have diffusion acting, and if you look at the influx phase only, so the first 0 to 14 hours on the X scale here, what we see is that the simulations, which is the solid line here, don't quite reach it up to the observed tracer. The blue one is in gray matter, the white is in, the green is in white matter. The dots are observations and the lines are simulations. So we can see that diffusion seems to play accounts for a substantial amount of the influx of tracer, but not quite. There seems to be, and similarly for the clearance phase, so from 10 hours and onwards, it seems that the clearance happens faster than what diffusion would predict. I'm sorry, I thought there was a question, but I realized it's the auto captioning here. Okay, so we can see that diffusion plays a role, but it does not explain the whole picture. What could other mechanisms be that could be involved? And really, with the first thing one could think of would be, well, how about if our diffusion coefficient is a bit wrong? We're taking it from images. We have this diffusion coefficient, anisotropic heterogeneous diffusion coefficient from the subjects, but what if it's off? What if really we should have a diffusion coefficient that is enhanced somehow by dispersion, for instance? Now we can model this by, instead of considering a diffusion coefficient D, we can multiply it by some enhancement factor alpha. We can test alpha 1, 2, 3, for instance. And what we then find is that we can either match the influx phase if we enhance the diffusion, but then our outflux phase, the clearance, is worse. So really neither this vanilla diffusion or an enhanced diffusion, whatever reason it could be, does not match the data well. So we can continue the story. We can ask, what about other modes of transport? How about convection? So in 1981, Helen Serre and her group indeed found a bulk flow of tissue fluid inside brain tissue, corresponding to around 5 to 15 micrometers per minute. So this is very slow, but it was consistently there. That's what they found. But just 5 years ago, Alan Berkman and his colleagues in San Francisco found no evidence for any form of convective transport in the brain. And so this community has really been, the discussion has been going back and forth for the last 40 years. And we, like Alice in Wonderland, have rather jumped into this rabbit hole in trying to explain this. But I think part of the reason for why there's this discrepancy and controversy in the experiments is that they're using the influx of a tracer, a dye, as proxy for measuring the underlying fluid movement, if existing. And it's an imperfect proxy. But at the same time, for us as mathematicians, this sets up a very interesting mathematical problem. So we can then approach this from a numerical or a technology perspective. We can ask, if we have these images of tracer movement or the tracer concentrations after 1, 6, 24, 48 hours, can we use computational techniques to solve the inverse problem, to do something about the underlying velocity field from them for those images? So that's the problem set that we're considering. We assume that we have 2 images, C1 at time 1, C2 at time t2, and we're interested in trying to identify a map that maps you from one image to the other in that time course. And it turns out that this is really a classical problem in computer vision that might not be very surprising. And it's often formulated as an optimization problem. So we're interested in minimizing an objective functional that quantifies the transport map. So for in this concentration is transported somehow via this velocity field phi that we're interested in computing. And we'd like this transport to be as effective as possible. And then we also want that this velocity field should not vary crazily. We have some kind of regularity. This is an optimization problem that can be solved via the Euler-Lagrange equations. And in fact, this is a problem that a version of this problem has been proposed for mice kind of contrast data at higher resolution. But we tested this approach and it really found that this was not viable here, partly because the transport here is transport only by convection alone. It ignores diffusion, which we've seen as an important component. And we also found that it was not robust for our purposes. We were unable to compute reliable estimates for kind of a range of numerical parameters. So we had to do something different. So what we did was a version of this, where instead of solving just the transport equation, and we decided that we're going to do something introduce a technique we call optimal convection diffusion. So we're still in the same setting where we have an image C1 at T1 and an image C2 at T2. Now we're interested in finding a velocity field such that this initial image convects and diffuses to the image at time T2 or almost to it, and also that this phi is reasonably regular. So after testing this approach, we found that this was surprisingly robust actually to our choice of numerical discretization, to our choice of regularization parameters. We can solve this in an iterative fashion. So we start with assuming that there's no convective velocity and simulate the corresponding concentration that would appear. And then we see how good is the mismatch or how good is the match with this mismatch at time t0. Then we adjust the velocity and solve this as an iterative optimization problem. And what we find is that with no convective velocity, we start at relatively large discrepancy, but as this optimization algorithm progresses, this objective functional is effectively reduced. We also find robust velocity estimates for numerical parameters. And you can see an illustration of the resulting velocity field computed for one of our sample patients here. This is in the clearance phase between, so a net velocity field between 24 and 48 hours. Now you may be thinking, well, this is very interesting and all, but were we really talking about sleep? Were we really talking about, such stuff as dreams are made on and the purpose of sleep? Well, here comes the twist to the story. So remember these patients, there's about 20 patients been rolled in and out of the MR machine. Well, about half of these patients, or 11 of these patients, they were in the hospital all day, but they were allowed also to sleep normally at night and continue their examinations and essentially just be a normally sleeping reference group. Then the other half of the patients, or more precisely around 8 patients, were sleep deprived. So they were in the hospital the whole first day, in and out of the MR machine. And then they stayed in the hospital. They were not allowed to sleep, again, in and out of the MR machine. And then they were released and were allowed to do whatever they wanted. And what our clinical collaborators, Computational and Geirigstad, who these data are the courtesy of, found was that the tracer clearance in these two groups differed. The sleep-deprived group, in red here, there the tracer lingered longer than in the reference group. And the clearance was lower by about a factor 2, which is very comparable to the original awake mice in Xi's 2013 science paper. Now, does our modeling reflect this? Well, what we could do is that we could estimate this average velocity field in the clearance phase, so between 24 and 48 hours for each patient. And then we can compute the average flow speed in the brain overall and in different brain regions. And what we found was that this convective velocity that we also estimated was also lower than by a factor 2 in the sleep-deprived cohort. So we persistently find flow velocities between 1 and 10 micrometers per minute, which is, we would say, surprisingly close to what Sir found in the 1980s in cats. And we find that it's substantially higher in the ones that were allowed to sleep normally than the ones that were sleep deprived. Now, you may ask, why are we really so interested in this convective flow and brain clearance? Well, there's two reasons why you're interested in brain clearance, and transport. And either you're interested in looking at how solutes move into the brain, like drugs, or you're interested in understanding how metabolic waste is cleared from the brain. And why are you interested in metabolic waste clearance? Well, because of neurodegenerative disease. So, as already was touched upon in the keynote just 2 talks ago, a number of neurodegenerative diseases are associated with this accumulation of toxic proteins in the brain, like amyloid beta or like the tau proteins. And whether one is interested or whether one thinks that these proteins are the cause of the disease or a byproduct of the disease, in any case, there's an interest in understanding how these proteins spread in the brain. And in particular, this protein hypothesis underlying, say, Alzheimer's disease stipulates that you have misfolded proteins in the brain tissue, and that these misfolded proteins, in a way, travel and lead to misfolding other proteins towards the brain tissue. And the current kind of gold standard for macro scale models of this protein interactions and propagation in brain tissue is based on these graph-based network models, that are represented by each region of the brain is represented by a node in a graph. And toxic proteins can either interact with each other locally, or can diffuse to other regions of the brain represented via edges of the graph. And these models have been rather successful actually in modeling how proteins spread, but they have two main problems. And well, there's probably more problems, but some of the problems are that they typically do not display the characteristic spreading patterns that's observed in some of these diseases. And also it really often the spread is too fast. There's not this long dormant phase that's often observed in toxic protein patterns. So we asked the question, how about the role of brain clearance in these models? This is something that is also not new. The role of brain clearance has been obtained a lot of interest in kind of neurodegenerative research, especially over the last 10 years, but it's not really been picked up in the model. So what we've done is that we introduced a new terms in these models for protein propagation, namely a clearance level. So we're now interested in modeling the propagation of the toxic proteins P and the clearance level lambda, and these can interact. So the clearance will affect the toxic protein propagation, and the toxic proteins will affect the clearance level. It's easy, I think, to understand the dynamics of the system if you just look at not a whole graph, but we just look at a single node. Then this becomes a 2x2 dynamical system for the concentration P and the clearance level lambda that has really two fixed points. Either there's a healthy state in which there's no toxic concentration, and the clearance level can be whatever it is, or there's a completely unhealthy state in which the toxic concentration is non-zero, it's at some limit level, and the clearance has been reduced to the minimal state. And this concept of a critical clearance, which is a parameter in a model, also defines a critical toxic load level. So if you start with a protein concentration that's lower than the critical one, the clearance will clear it away and you will end up in a healthy state. But if you start with a toxic level that's higher than the critical level, you will reduce the clearance to a level that is not, and it's unable to kind of clear the entire toxic load, and you will see accumulation of the disease. Now, of course, we can also look at this more at the network level rather than the single node. And what we find is that this concept of a dynamic clearance, it really affects the propagation of these proteins in our connectome model. It will delay the propagation of the proteins, as well as heterogeneous clearance maps will alter how the patterns and pathways for the spread. So with this, we can return to our original question in this context. So why do we sleep? Well, if we return to our cohort of sleep-deprived and sleeping subjects, what we can do is that in each region of the brain corresponding to our connectome, we can estimate the regional clearance rate. We're doing this by taking our signal intensity and concentration data at different times. For each region, we can estimate an exponential decay rate for the signal intensity in each patient. So this corresponds to, of course, to 1 night of sleep deprivation, and now we're extrapolating this for 40 years. But just for the example of demonstrating what could be the effect of this chronic changes, the changes you observed in 1 night of sleep if they were chronic. Okay, so based on these, we can create these individual clearance maps for each patient by taking this rate in each region. You can also look at the average clearance maps and the average regions. And what you see is that the average clearance level is really reduced after 1 night of sleep deprivation. This is not new. This is good because this means that the way we're computing these clearance maps reflect what is observed by the clinicians in their data. But now we can finally ask, we can take this 1 step further and see, okay, how about these differences in the clearance map? Will they affect the model for protein propagation? So in these experiments, what we do is that we trigger kind of the protein avalanche or cascade by placing a load of toxic chromosome in the entorhinal cortex node. And then we observe how the concentrations P and clearance levels spread. Here comes the question, where this runs. And so what you're seeing here are 3 sample brains. The top 3 are from our reference cohort, and the bottom 3 are from our now chronically sleep-deprived cohort. And you can see how the toxic proteins spread by the coloring. High is high levels of toxic proteins. You can see that there's large individual variety here as well. And there's clearly the lower 3 spread faster than the upper ones. This can also be reflected in differences in the Brock levels over time. So here we have kind of a modeling link between sleep and brain clearance at different levels, and brain clearance of the spread of toxic protein. So maybe that is why we sleep. Thank you so much for your attention. This is part of our work on modeling brain mechanics. I would be happy to talk to anyone here or at SFN about any of these topics. And I'd like to say thank you so very much to my very talented team, in particular my current or former PhD students, Georgia Brennan, Vegard Vinja, and Bastian Saff, Travis Thompson, Per Christian Eidengerdingstad for the data. Yes, thank you.
So much for that talk. Awesome.
Thank you. It's a great talk, very interesting. So a number of questions for you, but I'll focus on a couple dealing with this. Number one, in your modeling, do you take into account any intrinsic diurnal state?
Any intrinsic?
Diurnal, which is that it's not just sleep, it is the day, night, and the intrinsic cycling of the cells and cycling of genes involved in the cells.
No, there's no cells at this level.
I know,
but
but
macro scale.
in a higher level, do you take into account any such organization of
And so we're taking the data we have as a starting point with the confounders that are there, right? So it's 20 patients. They've done this over a time period of 48 hours, and we don't really know what they're doing after the sleep deprived, how they differ in the period from 24 to 48 hours, for instance, either. But they're all they're taking the same time of the day, so from morning to afternoon. And I don't think there's any, I think they all have kind of normal patterns for that matter. There's, yeah, there are no patterns.
And in terms of your convection,
yeah,
the protein, the change to the protein, I mean, the aggregation. Do you look at any local spatial organization of these protein clusters, or your clusters?
So there's two different directions there. So we're looking at just the convection aspect, which is on a whole different time scale than the protein modeling. So for the convection, we're looking at a tracer concentration at the macro scale, and you're looking at something that's been averaged over an MRI voxel. So that's kind of the scale we're looking at, right? And then also for the protein interactions, you're also looking at something that's on the scale of a brain region. So then we're at the centimeter scale. That's the road.
One more question.
Thank you. Also about the convection. What is the driving force for the convection?
Perfect question. So now I could answer this for hours. It's a standing question. I don't think anyone knows. There's hypotheses, some that make more biophysical sense than others. It's been proposed that there's some kind of, so brain tissue is heterogeneous and it's permeated by blood vessels. And there's a question whether is this convection happening everywhere, or is it happening mainly located in spaces associated with blood vessels? And the permeability of the extracellular space is very, very low. So theories that suggest that there is convection, significant convection in the extracellular space are unlikely. We don't consider that to be likely based on our findings. But there could very well be kind of lower resistance pathways along perivascular spaces. And one can imagine that the bulk flow that we observe here is really an average over a higher flow, but happening in smaller vessels. One can also observe that there are, one knows that there's intracranial pressure gradients in the brain of the orders of some micrometers of mercury per meter. But that these are, I don't know of any measurements in humans that are able to quantify something that's not pulsatile, that's not net zero. But what we know is that if you had steady gradients of the order that's been measured like a few micrometers per minute as steady pressure gradients, those would be sufficient to drive flow of the magnitude we see here. So that was a long answer to your question. But yeah.
Yeah, the question is, can we increase convection? Right? And
yeah.
What would that mean to a brain, as we've seen in his talk,
yeah.
where there's much more like unrestricted area around the brain?
So what's also interesting in our findings from this study is that if you're injecting tracer in the spinal canal, about 30% of that will reach the brain in the time course we see. So really what's also extremely important is the movement of the cerebrospinal fluid in spaces surrounding the brain. And there I think is partly it's easier to measure, but also there's larger velocities involved. I think that should also be an important thing to consider.
Well, thank you very much for that talk. I'm sure all of us who are old and sleep deprived are desperate to find out how to increase our convection rate. That's all I can think about your entire talk. Thank you so much. That was very interesting. We have a 10-minute break now, and let's try to get back here in 10 minutes. But I'm sure at this point everybody needs a little caffeine dose. I strongly suggest outside. There are 3 fantastic talks after the break, and then there is going to be a reception and a panel moderated by Roger Neal on autism. I think is very interesting. So please don't go away. Stick around. There's more